Welcome to this Tech Talk on how new standards, MDR and Brexit are having an impact on biocompatibility. I'm Lisa Arrigo with SAE Media Group, your moderator for this program. Today's speaker is certified microbiologist Thor Rollins, Director of Toxicology and E&L Consulting with Nelson Labs. Thor plays an active role along with the FDA and regulatory committees, developing standards, discussing biocompatibility methods, and voting on changes to those standards. During his presentation, you may download a PDF of his slides by clicking the Event Resources link on your screen, and you may send questions to him through the Ask a Question box. And now, here is our speaker.
Thank you, Lisa, and thank you, everyone, for attending today's talk. It's a great time to be involved with biocompatibility, although it can be a little frustrating, and that's what today's talk is going to be about. It's kind of perfect timing when you look at all the changes that have happened. So we have brand new ISO 10993 standards that have come out, and that's pairing with the perfect timing of MDR, which is basically hitting us right now as I'm giving this presentation. And also Brexit happened. And what that means from a regulatory standpoint is that while you're dealing with MDR, you have to think about the state-of-the-art standards that might have to impact your devices when you submit to your notified body for MDR. And at the same time, if you're looking to go into the UK, now you're dealing with a brand new regulatory department with Brexit. So you're kind of trying to deal with brand new ISO standards, which also are always frustrating. But then at the same time, you have your MDR and your Brexit happening all at the same time. And that's why we call it a whirlwind of this change. So we're going to cover very high level, the impact that we're seeing with these standard changes and how they correlate together with MDR and Brexit, so we see how we can pass through this the best way we can by saving as much money from testing and biocompatibility assessments. So first off, I'd like to say, why now? Like, why are we seeing these changes in biocompatibility? Why are we seeing these shortages of expertise and testing delays? And then why can't you just follow the new 10993 standard and go through regulatory submissions without any questions? These are things that I get all the time. And so to kind of get an idea of why we're seeing the changes, but also why we're seeing some of these impacts with expertise shortages and testing shortages, I'm going to talk about why what are these changes, what are happening right now in the industry. So traditionally, when we looked at biocompatibility, this is actually the old FDA's G95. It came out in 1995, and it was the standard for 20 years. On how to evaluate biocompatibility. This chart is from that standard, and it's a pretty easy thing to do. In fact, I joke, but honestly, it's not a joke. I taught my daughters when they were very early age how to go through this chart to be able to identify what biocompatibility tests were needed for certain devices. I think it's a really fun exercise. You look at your device, you look at the contact time to the body, you look at exactly where it's going in the body, and the length of time it contacts the body. And based on those determinations, it tells you what testing you need to do. And how we used to do biocomp was, quite frankly, we would just look at the chart and it would tell you what testing to do. You'd contact your favorite lab and you'd say, hey, I need to do these tests. And send me a quote and tell me how many samples I need. And that really was your biocompatibility. It was that simple. And so for the longest time, you had companies who would have engineers or QA people or maybe even my daughters do this exercise to determine what biocompatibility tests were needed for devices. And so it was easy, and it didn't really take a lot of knowledge about biocompatibility. It really just took this chart. Well, this is the way that we used to do things. The new way out of the new standards is this chart, and there's a lot of differences, and I'll kind of explain why, and the whole standard actually has a lot of changes, but this chart kind of sums up the majority of the changes in this short of time. So the first thing, as you can see, we added a whole category, a whole column to this chart, and that column is physical and or chemical information. And also, as you can see, this is the only column that we have an X in, which means it's the only column that's required for your medical device. So in the past, the other chart we showed, everything either had an X or O, which basically meant do the test. Here, we're seeing that the only thing that you're requiring to do for your medical device is to gather as much material information as you can. Then based off how the device is used and the risk and how much material information you have, you may need to do some chemistry to supplement that based on the risk of your device. After that exercise, you look at the information you gathered, be it material and or chemical information, and then based off the inputs of that material and chemical information and the risk of your device, you then evaluate the need to do the other tests. So it's really heavy on a risk-based approach, which is really the emphasis on the new 1093-1 that came out in 2018. It's more about risk, and the reason we did that is, one, medical devices are no longer just hunks of metal and plastic that we've used for hundreds of years. They're becoming much more complicated. We're seeing nanomaterials, we're seeing degradation materials, we're seeing combination products. And these tests have been around for decades, since the 50s, 60s, and 70s, and they were made to kind of gather confidence in these old materials, but they don't really work well with these new types of devices. So we had seen some issues from a biocompatibility standpoint on materials that pass these tests, quite frankly. So we started to lose confidence in the old checkbox approach. Plus, these tests are mostly done in animals, and we're trying to use less animals based on the risk of the device. So the concept here is if you look at the risk of your device, if it's a low risk device and you have lots of good material information and maybe even some chemistry information, then you can evaluate these endpoints and maybe not even have to do those tests. And this is what the benefit was of this risk-based approach. Was it was more based on the device individually instead of a general kind of statement. And so it did help us evaluate it a little bit closer. So as you can see, that's the other change, is we got rid of the X's on everything else and put evaluate. So that's the concept. You gather material and chemical information, you look at the risk of your medical device, and then based on the information you have about the materials, the processes that they go through, and the risk of the device, you evaluate the endpoints of every other needed test category. So that's the concept of the new 1093-1. New emphasis on a risk-based approach, increased emphasis on extractable, leachable testing, and so that's that chemistry information I was talking about, at least a portion of what could be chemical information. So lots of high-risk devices that maybe material information does not give you enough information to be able to do an evaluation. Then you add to that by doing extractable leachable testing, which is just looking at the chemicals that come off your devices and understand the toxicological risk from that. Now, the hard part about that, though, is this extractable leachable testing is not standardized. We're getting better. There's a new -18, 1093-18, that's just recently come out that really helps standardize more of this chemistry testing. But labs kind of do it a little bit different. And some labs that do it different still are very good, but there are some labs out there that honestly haven't quite met the requirements from the new standards. So that's part of the problem. The old animal testing that we're trying to get rid of, it's been around for so long that it didn't really matter if you went to lab A or lab B, besides some very few differences in how they were prepped, the samples, the testing was pretty much all the same. So the new extractable leachable testing is not standardized, so it does require expertise to be able to, one, understand what is needed, and two, to actually run the test. Also, there's a lot more emphasis on that initial risk assessment. We call them biological evaluation plans, or BEPs, but it's that initial risk assessment I talked about. So that's no longer my daughters that I showed a chart to, right? That actually takes someone who understands biocompatibility. If we're using chemistry, it takes a chemist. If you're using chemistry, you also need toxicologists to evaluate that. So we're starting to have kind of a group of people that need to do these risk assessments. You need biocompatibility people, you need chemists, you need toxicologists, and they're not always the same person. So sometimes you do need a crew of people to be able to do these types of risk assessments. We also have to consider cumulative exposure, which means, for example, if a device is used for an hour, only an hour on the body, but it's used every day for the rest of the patient's life. It's not just an hour exposure. You have to add up all those exposures, you do the math, and you determine what the total contact time is. And so that's part of this evaluation that needs to be done. And you're still looking at recently released devices, and you're looking at current standards for the MDR. So you might have a device on the European market forever, but now with the MDR, you have to reevaluate, does it meet the current state-of-the-art standards, which means have you gathered material or chemical information? So even if you have all that testing, remember the new standard, the only X is that material and chemical information. So you can have all the testing done, but you did not gather material chemical information, and there's a gap. So that has to be evaluated, and that's impacting with the MDRs. Now, this isn't just for MDR, though. As you can see here, this is actually right out of the FDA's guidance document for 10993, and it tells you in this paragraph, which is my favorite paragraph in the guidance document, exactly how to evaluate a medical device from a biocompatibility standpoint. And as you can see, it's kind of like a formula. The first thing you have to look at is the materials, that material information and chemical information that we've been talking about. You also have to look at the manufacturing processes. So a lot of materials are totally safe. Stainless steel, titanium, polypropylene, silicone, right? All these have been used for a long time. But a lot of times it's not the material that causes the toxicological concern. It's the residual processing, oils, detergents, colorants, things that you've either put in or put on the materials that actually becoming the risk. So you have to look at the manufacturing process. You have to look at the clinical use of the device. That's your formula. So, material components plus manufacturing process plus clinical use of the device equals the risks. So you look at that from a biological risk perspective, and then based off of that formula, you determine what testing is needed, either by testing, so a plan, either by testing or an evaluation for those endpoints. So once again, it doesn't mean you have to test. Even the FDA is saying if you can determine those endpoints by evaluation, that's the way they prefer it. So it's either test or evaluation. So this is the formula to do a risk assessment for the FDA. Very similar to what we talked about with ISO 10993. There's no surprise there. So as we've been talking, so you're looking at the new standard that just came out, ISO 10993-1. That new -1 has an emphasis on risk-based approach. That risk-based approach is heavily dependent on material and chemical information. So those are now a necessity when you're doing your risk assessment. So everything should have a biological evaluation plan performed, a risk assessment, looking at the materials and processes and the possible risk of the device to how it's used, and then come up with a plan to evaluate or mitigate those risks. This is how the standard is written. And if you are submitting under the MDR and you have missed one of these steps, even if you've done the tests, even if the tests were done 20 years ago and it's been on the European market for 20 years, all that doesn't matter. You still have a gap to the current state-of-the-art standard. And we're seeing that being asked from notified bodies under MDR. The other thing that just to keep in mind that the notified bodies are also going to require you to show some kind of educational or experience proof of that individual making the assessment. So it can't just be somebody who's doing a paper in your organization that really doesn't have the experience or education necessary to make these assumptions. So you have to be able to back up your biological evaluation plan with some kind of education or experience to be able to convince the notified bodies that this person is appropriate to make these assumptions. So that all plays into your MDR submissions with these new standards. The other thing that's made complicated is Brexit. So now that Brexit's happened, they are still accepting CE marks for now, but eventually the MHRA is going to come up with its own mark, its own application process or submission process. And in that case, you're going to have to, once again, have a new regulatory arena to submit to. And they're going to have their own kind of interpretation of this type of standard. I can tell you by talking to the MHRA, they're putting a lot more emphasis on that initial chemistry to understand risks. They're really trying to push their submissions to the point where you do your chemistry to really understand. So it's not like you can do chemistry and then do the testing too and do it side by side and in parallel. They are holding to the spirit of the standard, which I love from a scientific perspective. It does put a little bit more time in the essence, but the concept is you do the chemistry and your material characterization, and that really makes you drive to what testing is needed. So the chemistry and material tells you what to do, and that expertise has to be there. So those are the two things we're seeing from Brexit, from MHRA, is more of an emphasis on the upfront material gathering and the expertise needed for it. So Brexit is happening. You have to consider how MHRA is going to interpret the standard. MDR, once again, is brand new, so the notified bodies are looking at these new standards as state-of-the-art and filling in the gaps. And how we look at saving money for all of this is actually using the right education and experience on biocompatibility assessments to determine what testing is even necessary. So when you're looking at the testing evaluations that we talked about before, the secret to this whole concept, even though it sounds like it's so much harder, so much more onerous on you as far as a medical device manufacturer, you got to remember that if you do this correctly and you gather the right information up front and even to do the chemistry, we're able to evaluate a lot of those biological endpoints from a risk-based approach, which means you're not spending the time for, the 16 to 19 weeks it takes to do an implantation test. We may not have to do an implantation test, depends on the material information you gather. Or the subacute subchronic toxicity, or even worse, carcinogenicity evaluation. Those endpoints under this risk-based approach, we may be able or should be able to assess a lot of those unless you're using really weird materials through a risk-based approach. So even though it sounds much more complicated on paper, and especially from someone who's used to a checkbox mentality to try to do a risk-based approach looking at chemistry and materials, the truth of it is if you do it right, you can spend a lot of time and a lot of money by justifying out of testing if it's done correctly. So that's the benefit of this whole approach, keeping in mind the different regulatory arenas you're trying to get into. That plan should really, if you're looking to go to Europe, if you're looking to do FDA, if you're looking for Canada, if you're looking for Asia, if you're looking for the UK, then you really want to develop your risk-based plan for all those regulatory bodies. And so if you can do that effectively, then you really do end up saving a lot of time and money in the back end. But a lot of times that's a lot of more upfront work that's needed. Well, I appreciate all the time that you've spent. We didn't have a whole lot of time to go into this in depth, but I'm hoping that you were able to gather a lot of information from the high level we talked about. But I want to thank you for your attention, and now I'll turn the time back over to Lisa. Lisa?
Thank you, sir. That was a wonderful presentation. This Tech Talk will be archived on our website for the next 12 months. Thanks for watching.